I've been asked to give an overview of what immunotherapy for cancer is, and I think that in order for you to understand what cancer immunotherapy is, it's easier to understand how the immune system works normally. So I'm going to talk about some of the players in normal immune function, and then I'm going to talk about types of cancer immunotherapy and how they relate to the normal functions that we discussed earlier, okay? So probably the first and most famous player in this game is the monoclonal antibody. So antibodies are part of your immune system. B cells make them, and they recognize viruses and bacteria that you've been exposed to. Antibodies have two parts. They have what's called the antigen binding fragment. So antigens are bits of protein that are recognized by the immune system. Each antibody usually has two. And then there's a third part, a tail called the constant fragment, or FC. And the FC is also an important part of the structure of the protein because it mediates functions like how antibodies talk to other parts of the immune system. The next component that I want to talk about is the cytotoxic T lymphocyte, or the killer T cells. T cells have a number of important proteins on their surface which determine how they interact with both normal cells, infected cells, and cancer cells. The most prominent of which is the T cell receptor shown at the top. The T cell receptor also recognizes antigens, but it recognizes it in a slightly different context, and I'll discuss that in a little bit more detail. In addition, there's a number of surface molecules on T cells that help turn it on or turn it off. These are activating co-receptors such as CD28, and this is one of the turning on signals. And there's inhibitory co-receptors such as CTLA4 and PD1, and these turn off immune responses in the T cell. And another important cell that I want to talk about is natural killer, or NK cells. Natural killer cells have important functions in which they can directly kill either infected cells or cancer cells. And they do this because they can express on their surface an FC receptor. So you saw earlier in the antibody structure that they have an FC, or constant region. And the FC receptor will bind to antibodies that are, for example, stuck to the surface of infected cells. And we'll talk about that in a moment. So under normal circumstances, if you have a normal cell, normal cells are constantly recycling bits of normal proteins that they make, and they're recycling them onto the surface of the cell in something called the major histocompatibility complex. So the MHC is a way for the normal cells and cells in your body to communicate with T cells. And if you've ever heard about tissue typing, what they're talking about is major histocompatibility typing. So in this cartoon, you can see that on the MHC, there's little red, blue, and yellow bits of self proteins, or self antigens, which are normal, and they should not provoke an immune response. So cytotoxic T lymphocytes, or killer T cells, they circulate around your body, and their T cell receptors will look at the major histocompatibility complexes that they encounter. So if they read the protein that's stuck in the major histocompatibility complex, sort of like a barcode, and if they see that it's a self protein that is normal and it's happy to see it, then there's no positive signal. In addition, normal cells express inhibitory receptor ligands such as PD-L1. These are the so-called immune checkpoints that you hear so much about. And that's an additional negative signal to prevent the cytotoxic T cell from being activated. So if it sees something like PD-L1 on the surface of a normal cell, it's another red light. So there's multiple safeguards in your immune system to prevent activation against normal cells. So what happens when a cell gets infected with a virus? So here's a virus, it gets into a normal cell. Viruses hijack your cell's protein production machinery and uses it to replicate themselves. And they'll make virus proteins. And some of these virus proteins will end up on the surface of the cell in the major histocompatibility complex. Now, a cytotoxic T lymphocyte is circulating around, In addition, inflammatory conditions sometimes will downregulate some of those negative signals on normal cells. So there goes the PD-L1. Now the cytotoxic T lymphocyte scans the MHC complex and it sees a virus protein that it doesn't like. And so that's a go signal to kill that cell. In addition, there's no negative signal to downregulate the activity of the T cells, and so it will kill the targeted cell that's been infected by the virus. Now, how do antibodies contribute to these functions? So if a virus infects a cell, some of those virus proteins are going to end up on the surface of the cell as well. And these are whole proteins that can be recognized by antibodies. So these are non-self proteins on the surface of the cell. Antibodies can see them and stick to them. So the antigen binding portion of the antibodies, as you can see in blue, are sticking to the virus proteins. And then an NK cell, which expresses that FC receptor, is going to go in and bind to the FC portion of that antibody, and it will kill the infected cell. So those are normal immune functions. That's what happens every day. You get infected with a virus, or you have a cold, or you catch a bug. This is what happens in your body. So cytotoxic T lymphocytes normally survey the self for non-self antigens, and they'll kill infected cells. And antibodies also stick to cells or proteins that express non-self antigens, and they mark them for immune destruction. Now, there's a number of different immune therapies in cancer which try to leverage those normal functions and turn them against cancer cells. And I should preface this by saying it's pretty clear now that under normal circumstances, some people's immune system will mount an immune response against cancer. But it's very rare that those anti-cancer immune responses are going to protect them, and we'll talk about why at the end. So a number of immune therapies are either in investigation or have been approved by the FDA. So there's targeted monoclonal antibodies. So these are antibodies that have been engineered in a laboratory to recognize tumor-associated targets or to modulate immune responses. There's tumor vaccines, which, like infectious disease vaccines, are designed to train the immune system, and particularly T cells, to recognize and kill tumor cells. There's engineered T-cell therapies, which have been getting a lot of notice lately, which are very interesting. And these are engineering T-cells from individuals by adding a receptor, or an engineered receptor or a T-cell receptor, in order to specify that those T-cells will target tumor cells specifically. And then there's checkpoint inhibitors, which are monoclonal antibodies that are designed to block or modulate inhibitory receptors and signals to the immune system. So what are these things? So targeting monoclonal antibodies are targeted against surface proteins that are expressed in tumor cells. So in my field, multiple myeloma, myeloma cells, which are cancers of the immune system, they express a marker on their surface called CD38. And there's a lot of interest now. There's an FDA-approved antibody called daratumumab, which recognizes CD38. So in this picture, you can see that the myeloma cell expresses CD38 on the surface. Daratumumab sticks to the CD38 and attracts natural killer cells to the tumors, and those natural killer cells can kill the myeloma cells. So targeted monoclonal antibodies are targeted to proteins or antigens that are expressed by the tumor cell. And that's a little bit different from the checkpoint inhibitors, which we'll talk about in a moment. So tumor vaccines are designed to train your immune system, to train your T cells to recognize cancer cells. So in this context, we will try to isolate proteins or antigens that are relatively specific to tumors. In this example, myeloma cells express a protein called MAGE-A3, which is relatively specific to the cancer. So we can make vaccines out of MAGE-A3 or other tumor-associated proteins and use them to vaccinate patients who have cancer. And those patients will mobilize immune responses from T and B cells. And the T cells, as depicted in the right hand of the cartoon, MAGE-A3 specific T cells can kill tumor cells that express MAGE-A3 and have it on their surface. And in addition, MAGE-A3 specific antibodies can help bind up that tumor protein and amplify the immune response. Engineered T-cell therapy takes this one step further by taking peripheral blood from patients who have cancer, isolating their T-cells, and then putting a gene into those T-cells so that those T-cells will express a new receptor. So sometimes that's a T-cell receptor gene, which is specific for a tumor antigen, such as MAGE-A3. In other cases, there are what are known as CAR, or chimeric antigen receptor, which confer on the T-cell specificity for a particular protein that's on the surface of the tumor cells. And those engineered T-cells will then express those receptors on their surface, and then they're reinfused back into the patients. That's what's called adoptive T-cell therapy. And then those engineered T-cells will seek out and kill the tumor cells. And finally, immune checkpoint inhibitors are the best studied and the most broadly available immune therapies that are available now. The way that immune checkpoint inhibitors work is, as we saw earlier, cytotoxic T lymphocytes scan around the body and looking at the MHC, looking for tumors. A tumor-specific T cell is looking for a tumor-associated antigen. And so it reads it like a barcode, and it sees that it's something that it doesn't like, and that's a go signal. However, a lot of tumor cells express inhibitory receptors such as PD-L1 on their surface, and those will bind to PD-1 on the T cells, and that's actually a red light. It turns down the immune response. So even though that T cell wants to kill the tumor cell, a negative signal is putting the brakes on the immune system, preventing those T cells from effectively killing the tumor cells. So that's why even in patients who do have immune responses, they don't necessarily get protection from them. What immune checkpoint inhibitors do is they block those interactions, those negative signals. So the checkpoint inhibitor antibody can stick to PD-1 or PD-L1, preventing that interaction from occurring and preventing that negative signal from turning off the T cell. And so that T cell can go ahead and kill tumor cells. So all of these immune therapies fit into the context of what we call the cancer immunity cycle. So it takes a lot of steps, just like when you get an infection or if you get vaccinated to the flu or pneumonia or what have you, there's a lot of steps in the generation of an effective immune response and to keep that immune response going in order to effectively eliminate the infections. And in the same respect, there's a lot of steps that go into anti-tumor immunity. So there's things that happen at the tumor site, and this diagram depicts these critical interactions that happen either at the tumor site or when the T cells go into lymph nodes or when those T cells go back to the tumor site to kill the to kill the tumor cells. So many of the immune therapies that we just discussed only act at one or two of these critical points. So they're dependent upon the other steps in the cycle being in place already. So the challenges that face immune therapy now in terms of targeting monoclonal antibodies, we want to find better cancer-specific targets or new targets for cancers that we currently don't have antibodies available for. For tumor vaccines, again, we need to find the best antigens and the best vaccine formulations in order to generate broad and deep immune responses. Engineered T cells, we're still looking for targets and T cell receptors for specific types of cancers and different types of targets. And checkpoint inhibitors, what are the best inhibitors and when should we give them? And ultimately, I think the future lies in combining these different therapies, because if we can put the right combination into every point in this cancer immunity cycle, it will give us an opportunity to generate broad, effective cancer immunity for patients with many different kinds of cancers. And so we will have higher response rates and better responses in patients with cancer. So our challenge now that we have made the important first step of bringing these immune therapies into the clinical setting is to now understand how we combine them. What do we put them together with? What different immune therapies can put together? Can we put these immune therapies together with conventional chemotherapies? And are there other therapies, such as therapies that are used typically to modulate immune responses, that will make these treatments more effective? And with that, I'll close, and I'll be happy to take any questions from you right now. Was that clear? Yes, sir.
Is there risk at all in potentially entering into a clinical trial that would both be providing both the traditional chemo regimen plus immunotherapy? Is there risk in providing the two together to a newly diagnosed patient, I guess is the question. Because one is cytotoxic systemically and the other one is more targeted, and is there concern about the two, or does that sound like a reasonable first step given the fact that you're actually going to at least get the traditional therapy and maybe also get the benefits of immunotherapy?
So that's an excellent question. Just to reiterate, the question is if a newly diagnosed patient has an option to go on a clinical trial that combines both traditional chemotherapy and immunologic therapy, are there any risks? Well, that's an excellent question. At the moment, all the cancer immunotherapies are approved for relapse or progressive disease. There's also specific situations for what's called adjuvant therapy. But there's a lot of interest in bringing them to the frontline therapy to both improve response rates and increase the durability of responses. But these combinations are investigational, they're experimental. So there's always going to be risks. The immune therapies themselves have certain risks. By activating the immune system, sometimes you can cause autoimmune functions. You can have recognition of self-antigens. Any specific immunologic therapy is going to have risks. So the short answer is yes, there is risk. Another more subtle question is, can one type of therapy potentially negatively impact another therapy? For example, are conventional chemotherapies immune suppressive, and therefore are they going to negate the effectiveness of an immune therapy? Well, the answer is there's evidence for both, because in order to generate an immune response, you have to kill some tumor cells first. And it turns out that certain types of chemotherapy will synergize. They'll work together with immune therapies to generate immune responses. So in general, I can't speak to a specific trial that you may be thinking about, but in general, the effort is to design complementary therapies. So chemotherapies that will not negatively impact immune response, but will help generate immune responses by various different actions, and then putting them together with immune therapies that should amplify those responses. But there are risks. There's always certain risks specific to different types of therapy. Even every FDA-approved drug, if you ever look in the package insert, there's a whole long list of potential side effects. And when you're talking about cancer therapy, the stakes are much higher. You really have to think about that. So if you do speak to a clinician about a clinical trial, you really should ask your questions about what are the risks, what are the potential benefits. It's very important. Yes, sir.
I was wondering, with respect to augmentation with different immunotherapies, are there clinical trials that are using more than a single immunotherapeutic approach?
So the best studied combinations so far are different combinations of those checkpoint inhibitor antibodies, which have proven to be effective, and there's an FDA approval for that. But I think the next step are clinical trials combining either chemotherapy approaches that affect parts of that cancer immunity cycle, plus immune therapies that should affect other parts of the cycle, or combining different immune therapies that should affect different parts of the cycle and amplify the immune response. So I think the next generation of combination clinical trials are along those lines. So there's different combinations of antibodies, for example. And in my field, in multiple myeloma, there's a number of clinical trials underway combining targeting antibodies, which are directed against the tumor cell, with checkpoint inhibitors, which are directed to amplify the immune response. Yeah.
Can you talk a little bit about having stem cell transplant and how immunotherapy is combined with that, or can work with that, or you wait to use that down the road? how that would affect overall remission.
Right. So the question is how stem cell transplant can be combined with immune therapy, if I'm understanding. So autologous stem cell transplant is a type of therapy that is used commonly in blood cancers. So in diseases like lymphoma and multiple myeloma, there's a very well-defined role for what's called autologous stem cell transplant. So autologous stem cell transplant is not a transplant in the conventional sense. So in what's called an allogeneic transplant, which is what most people think about when they think about transplant, bone marrow stem cells are taken from a donor and given to a recipient. And that's actually immunologic therapy because the new immune system that grows from the donor cells recognizes the cancer as being foreign and it kills it. And that's been well demonstrated at a cellular level. So for diseases like leukemia, an allogeneic stem cell transplant from a donor is curative, can be curative. In other diseases, such as lymphoma and myeloma, we use what's called an autologous stem cell transplant. Autologous stem cell transplant isn't really a transplant. We take stem cells from the patient, and then we can give very high doses of chemotherapy, and then we give those stem cells back so that the bone marrow can recover and the immune system and the blood-forming system can recover. And it's really a way to support very high doses of chemotherapy, but it has certain effects that may be beneficial because those stem cells have to re-engraft and they also make new immune cells. Now, as of now, there's no evidence that new evolving immune system is going to recognize cancer cells with any greater frequency than they did before. But there's a lot of interest in applying immunologic therapies after transplant or around the time of transplant in order to shape that new immune system coming back in to kill cancer cells. So there's efforts in myeloma to use vaccines, to use targeting antibodies, and to use immune checkpoint inhibitors after the transplant to help direct the immune response, the immune system as it regrows to kill cancer cells. So that's a research effort. We don't have any FDA-approved therapies for that application yet, but it is a big area of investigation, and there's a lot of interest in trying to do that. In other words, to get the allogeneic, the donor transplant experience for patients who undergo auto transplant. I think we're out of time now. So thank you very much for your attention. I really appreciate it.